All right, friends, welcome back. We are coming down to the end, aren't we? There's like single-digit number of class periods left, a couple of weeks, and then we're going to be done. So between here and there, we got quite a bit to cover. We're going to spend today on DNA and RNA, nucleotides, and so on. Next time we begin metabolism, we'll talk about what happens to lipids, a little bit about what happens to proteins, not too much, and we'll talk about what happens to carbohydrates. And then we'll have one last chapter, probably 28, on organometallic chemistry, so going right back into reactions, and that'll probably take us through the end. So today I think will be easy for a lot of you. If you've had Bio 240, you probably know a lot of the stuff already. So on the other, I worry about that a little bit because I don't want to spend too much time on stuff you already know. On the other hand, somebody once told me about scientific presentations that you shouldn't worry about telling people things they already know. It helps them feel smart, and they like that. So this may be a good day at reviewing things, and hopefully we'll talk about some things with a slightly different perspective than you've heard before. So you can group the nucleobases, meaning the heterocyclic portion of DNA. The term heterocycle refers to a ring that contains nitrogen or oxygen. So you'll sometimes hear people talk about heterocyclic bases. They come in two flavors. One is called purines, and the other is called pyrimidines. And those names are not sure what the origin is of it, though I do know that there's a certain kind of organic chemist that really enjoys being able to say the complicated, non-standard name of a aromatic ring that contains nitrogen/oxygen. It's how we sort of impress each other. It's the OChem flex. For example, isoxazolidenone, that sounds kind of cool to say. And if you can say it without looking like you're thinking about it, that's even more impressive. Purines and pyrimidines are less impressive versions of this. It's useful to group the bases in these categories because later on, naming them, their names will be a little bit different depending on whether they're purines or pyrimidines. So the purine bases are the ones that are larger. These have a 5-membered ring plus a 6-membered ring fused together, whereas pyrimidines are just 6-membered rings. So the two purines have the same basic structure in the sense that the positioning of carbons and nitrogens in the ring is going to be identical. So here's the 5-membered ring, and then here's the 6-membered ring, and they always have nitrogens at those positions. The nitrogen on the bottom of the 5-membered ring is always the one, the squiggly line indicates that this is where the attachment point is to the sugar. There's always a double bond here and here, and then the 6-membered ring is different depending on whether it's guanine or adenine. If it's guanine, you have pi bonds involving both of those nitrogens, and then you have an NH2 group here. This purines actually and pyrimidines together become a useful way to test whether you still got it when it comes to aromaticity and which nitrogen is more basic. For example, this nitrogen, you expect the lone pairs to be in an sp2 hybridized orbital orthogonal to the pi system of the ring. Why? Because that nitrogen is already involved in a pi bond, so you'd expect this nitrogen on top to be basic. The nitrogen on the bottom you expect to not be basic because its lone pairs are part of the aromatic system. 2, 4, 6, 8, 10 electrons, so it's aromatic. The other two nitrogens that are part of the rings, again, the lone pair should be in an sp2 orbital. The lone pair on this amino group that's outside the ring should be in a p orbital where they can participate in conjugation. We would expect this nitrogen to be not very basic, again, because of conjugation of the lone pair with the ring. This nitrogen that is outside the ring is called an exocyclic amino group, and several bases have them, just in different positions. So I'm going to refer to them as exocyclic amino groups or exocyclic amines. The other thing we'll do is we're going to highlight the groups that can be hydrogen bond acceptors, meaning electronegative heteroatoms with lone pairs that can interact with hydrogen, and then hydrogen bond donors, which are functional groups that have a bond between hydrogen and an electronegative heteroatom such that the hydrogen is electron poor. So acceptors will be blue, donors will be pink. Notice that adenine has donors here, here, and here, and an acceptor there. The lone pair on the exocyclic amino group is not a hydrogen bond acceptor because it's tied up in resonance with the aromatic ring. Okay, questions about that? Yeah.
Was the exocyclic group a donor?
It is a hydrogen bond donor. Yeah. The exocyclic amino group is a hydrogen bond donor. It is not a hydrogen bond acceptor because its lone pair is not basic. It's tied up in resonance with the ring. In my mind, I often get the words donor and acceptor when it comes to hydrogen bonds mixed up, and here's why. I think of nucleophiles as being donors of lone pairs and electrophiles as being acceptors of lone pairs, which is exactly backwards when you talk about hydrogen bond donor versus acceptor. So I'll try to keep that straight. Other questions? Yeah?
Why is the exocyclic amino group a donor but not the one in the ring itself?
So the exocyclic amino group is a hydrogen bond donor because it has these bonds between the nitrogen and the hydrogen that are the electron poor hydrogen. Yeah. Whereas the ones in the ring don't have bonds with hydrogen. Yeah. Others? Okay, so let's do guanine, and then we'll do the pyrimidines. So for guanine, you're going to keep the same arrangement of nitrogens in the ring. But unlike adenine, guanine's going to have a carbonyl on the top edge. It's going to have an NH here, and it's going to have an exocyclic amino group here. Other than that, we'll keep everything else basically the same. Looking at where the lone pairs are in guanine, you have an exocyclic amino group that is a hydrogen bond donor. The lone pairs are not hydrogen bond acceptors because they're tied up in resonance with the ring. Same thing with this nitrogen. That lone pair is not basic. It's not a hydrogen bond acceptor because it's tied up in resonance with the ring and with the carbonyl. So these are two hydrogen bond donating groups. You have electron poor hydrogens bonded to electronegative nitrogens. The carbonyl oxygen is a hydrogen bond acceptor, so is this nitrogen, and so is that nitrogen. So you can see that guanine has more groups capable of hydrogen bonding on this sort of right-hand side of the ring. Okay. So now let's draw the pyrimidines. The pyrimidines are all six-membered rings. The first is called uracil, one letter code U, and all of the pyrimidines are going to share this arrangement of nitrogens. The one on the bottom is the one that's attached to the sugar. They're all going to have this double bond, and then they'll have different groups on the right-handed side. For uracil, you've got two carbonyls and an NH. The nitrogen lone pairs are not hydrogen bond acceptors because they're tied up in resonance with the ring, so this NH is a donor. The two carbonyls are acceptors. Uracil is found, as you know, in RNA. Thymine is found in DNA. And thymine differs from uracil only in the presence of a methyl group. So you can copy down the structure for thymine exactly, and thymine just has a methyl group there. And you'll notice that the pattern of a hydrogen bond donor acceptor in adenine from top to bottom on this edge mirrors in an inverted way the pattern of acceptor donor on uracil and thymine. So those two are complementary to each other in Watson-Crick base pairing, and I'll show you what that looks like. The last pyrimidine base is called cytosine, and this is a potential point of confusion. I wish they'd chosen a different name, but they didn't. You'll see later on that the nucleoside version of these bases, when they're attached to ribose sugars, are adenosine and guanosine, but what the word cytosine means just the nucleobase for C. So it's a potential element of confusion. I want to make sure you memorize it right. Here is the same six-membered ring structure. The nitrogen's in the right place, double bond here. Cytosine has an exocyclic amino group and then a carbonyl, and the nitrogen in the middle is a hydrogen bond acceptor. Lone pairs on the exocyclic amino group are not basic because they're tied up in resonance with the ring. So you can see that cytosine has a hydrogen bond donor and then two acceptors, which is complementary to the acceptor and two donors that is on guanine. Okay, so you should have those structures memorized. You should be able to recognize them, draw them. You should be able to say what their correct names are. You should expect to find uracil in RNA ribonucleosides. You should expect to see thymine in deoxyribonucleotides and nucleosides. Okay, you guys are familiar with Watson-Crick base pairing. You've seen that before. Can you draw it? Yeah, go ahead.
A, T, G, C.
Right, A and T pair together, G and C pair together. A and U also pair together, though that would be in, U would only be an RNA strand. So you would see AU pairing either within RNA or in a hybrid structure between RNA and DNA. So let's look at that Watson-Crick base pairing. Let's look at an AT pair. So we'll draw A on the left. Then there's a hydrogen bond between the exocyclic amino group of A and one of the carbonyls of T. The methyl group of T is here. Attachment to the sugar is there, and then there's another carbonyl for T there. And this dotted line here indicates a hydrogen bond. So, hydrogen bond acceptor pairs with hydrogen bond donor. Similarly, hydrogen bond donor on T pairs with hydrogen bond acceptor on A. Notice that there are still other functional groups in the ring that are not involved in Watson-Crick hydrogen bonding. You've got these hydrogen bond acceptors here and here, which are unsatisfied. In double-stranded DNA, so this is an AT pair, this is A, this is T. In double-stranded DNA, this portion here is exposed to the major groove. This edge of the base pair is exposed to the major groove, whereas this portion here is exposed to the minor groove. That's important, and we'll come back to it, but the idea is if you look at the major groove of DNA, you should expect to, for an AT pair, you should expect to find a methyl group a carbonyl oxygen, an exocyclic amine, and then this ring nitrogen from the purine. And that'll be distinctive for an AT pair, and we'll talk about that in a little bit. Let's do the same for a GC pair. Notice that the AT pair has 2 hydrogen bonds, 2 intra-strand hydrogen bonds. In contrast, a GC pair, as you will see, has 3. So here's the purine ring for G. Nitrogen. For G, you've got the carbonyl oxygen here. Got a donor here, and then you've got the exocyclic amino group here. The carbonyl oxygen is hydrogen bound to the exocyclic amino group of C. I always struggle with the structure of C more than the structure of T. All right, so the carbonyl oxygen here, hydrogen bonds with the exocyclic amino group of G. The ring nitrogen, hydrogen bonds with a ring NH in G. Then the exocyclic amino group of C, hydrogen bonds with the carbonyl of G. So this is the GC pair. You can see the 3 hydrogen bonds. And that plays a role in molecular biology. If you've ever worked in a molecular biology lab or done some work with DNA oligonucleotides, it's often important to know whether your DNA sequence has a lot of GC pairs versus AT pairs. The increased number of hydrogen bonds for GC pairs versus AT pairs means that oligonucleotides that are rich in GC pairs hold on to each other more tightly than those that are rich in AT pairs. And that plays a role in how you calculate what annealing temperature you want for a PCR reaction. If none of those words sound familiar, don't worry, you'll learn more about it in biochem. If I tried to explain it, I'd be embarrassed because I'd get some stuff wrong. But all right, any other questions about Watson-Crick base pairing? Yes, go ahead.
So, as I recall from my biology classes,
Oops.
there's like they kind of go, the pair like has their flip on the head, like since both strands go in different directions.
That's right. We'll talk about double-stranded DNA. They're anti-parallel to each other. We'll talk about the 5' end and the 3' end. Good. Yes?
Can you explain major and minor group a little bit more?
Sure. That won't become clear. Why don't we just jump there now, if that's okay? We'll have a bit to say about nucleoside versus nucleotide structure. Just as a preview, a nucleoside has the base plus the sugar. S stands for sugar. Nucleoside. Nucleotide has 3 things. T is for 3. It has base, sugar, and phosphate. DNA is an oligonucleotide. You have lots of base, sugar, phosphates linked to each other as a polymer. And 2 oligonucleotides associate with each other to form a double helix, which you've probably seen before. I'll just show you an image of that. Before I do, this side of the base pair in a double helix is again, the top side is the major groove, the bottom side is the minor groove. And I'll show you what I mean by that. So here is, why don't we just, I didn't do this last time, probably should have done. Copy. There we go. Okay, so here is an oligonucleotide strand. Here is the deoxyribose ring. It's called deoxyribose because it lacks a hydroxy group at the 2' carbon, which is where ribose would be. By the way, this issue of 5' versus 3, I am going to get to the question about the major and minor groove. I'm just trying to orient you to the way things are. The deal with the 5' and 3' numbers is we number the atoms of the purine and pyrimidine bases with 1, 2, 3, 4, 5, 6, 7, 8, 9 as appropriate. To distinguish, we number the atoms of the ribose ring with 1', 2', 3', 4', 5'. So this is the deoxyribose ring. If it were ribose, there would be a dash OH at carbon 2. In an oligonucleotide, the 5' hydroxyl group has been modified with a phosphate. The 3' hydroxyl group has been modified with a phosphate, and that phosphorus is connected to another 5' hydroxyl group. That linkage, a linkage with an alcohol and a phosphate on one side is called a phosphoester. If you got two of them, it's a phosphodiester. So you've probably heard about the phosphodiester linkage between nucleotide units. So in an oligonucleotide, oligo means many, nucleotide is the base, the sugar, the phosphate. An oligonucleotide is many nucleotides linked by phosphodiester linkages. You have what's called the sugar phosphate backbone, and then you have the bases. One strand of DNA, as shown here, will pair with another strand. Like peptides, really, DNA strands have directionality. There's a 5' end and a 3' end. You probably heard about that. That just refers to the sugar. So the 5' hydroxyl's on this end, the 5' phosphate, and then the 3' hydroxyl. When you go to make a nucleotide, which I will show you the reaction for that in a little bit, the 3' hydroxyl is the nucleophile. So with that in mind, you can imagine a very long DNA chain, and here is how the double helix forms. The 5' end of one strand going to the 3' end of the other, and then that pairs with a complementary strand, but in an anti-parallel direction. So 5' on this end and 3' on that end. So do you see the anti-parallel orientation of those two strands? Okay. That's double helix DNA, and if you look at that from a space filling point of view, you get this. So here are the two strands, and you can see the sugar phosphate backbone. Just to get you oriented, in this space filling model, all the carbons are gray, hydrogens are white, oxygens are red, nitrogens are blue, phosphorus is yellow. So you see in double-stranded DNA, you have this, what's called the sugar phosphate backbone on the outside. You can't totally see the ribose rings. You can certainly see the phosphate groups, phosphate, deoxyribose ring, phosphate, deoxyribose ring, and so on. So this is one of the strands, and then this is the other strand. And you can see in a double helix, there's alternating spacing between sugar phosphate backbones. There's a narrow, deep spacing here, and then a broader, wider spacing here. The narrow, deep spacing is called the minor groove, and the broader, wider spacing is called the major groove. And the reason that's important is because there are things called transcription factors, which recognize sequences of DNA based on well, we'll talk about what that's based on. They recognize sequences of DNA. They recruit RNA polymerases to the DNA. They spread the DNA strands apart and allow for an RNA strand to be transcribed from the DNA. So that's important. That's an important first step in turning on a gene that allows that its gene product to eventually turn into RNA and then DNA. The place where a lot of transcription factors bind is here in the major groove. Yeah, go ahead.
Why are major and minor grooves a thing?
When you have the two DNA strands associate with each other, they form this helix, and they found that the sugar phosphate backbones. So why is it a thing? I would say the geometry of the oligonucleotide makes it that way, though I realize that's not a satisfying answer. You have one side where of this twisting double helix where the spacing between the two sugar phosphate backbones is close, and another side where the spacing is farther away. And again, I don't totally know why it's that way. It likely has to do with particularly the rotation around the bonds in the sugar phosphate backbone, probably some conformational properties of 5-membered rings. The thing that probably determines it is the sugar phosphate backbone has to adopt whatever geometry facilitates optimal Watson-Crick base pairing between the bases. Okay, so I want you to notice that the minor groove is narrow and deep. The major groove is broad and wide. Straight is the way and narrow the gate that leads to the minor groove. And broad is the gate and wide the way that leads to the major groove, and many transcription factors go in there at. See, you can. This is we're mingling organic chemistry with scripture. We're sorry about that, but there we go. So I want you to notice the different kinds of functional groups that are displayed on an AT pair versus a GC pair. Okay, AT pair you expect to see a methyl group on the T side, then a carbonyl oxygen, and on the A side you expect to see an exocyclic nitrogen and a ring nitrogen. Methyl, oxygen, exocyclic amine, ring nitrogen. For a GC pair, on the C side you expect to see exocyclic amine, then on the G side carbonyl oxygen, then ring nitrogen. So let's see just by looking at the minor groove edge whether we can identify an AT or TA pair versus a GC or CG pair. We want to recognize the pair. We want to be able to tell which base is on the left versus which is on the right. Because if we can do it with our eyes, then a transcription factor can do it with functional groups, recognizing hydrogen bonds. So here we go. Here is the major groove. Let's focus in on, just briefly, this base. I'm putting it in a box. All right? We're looking at the major groove edge. There's a methyl group. There's an oxygen. There's an exocyclic amino group, and there's a ring nitrogen. So I'll label those here. Here's the nitrogen, the ring nitrogen with a lone pair on it. Here's the exocyclic amino group. Here's the carbonyl oxygen, and there's the CH3. See that? Now if we go up to our drawing of Watson-Crick base pairing, that's too many pages away. Methyl, carbonyl oxygen, exocyclic amine, ring nitrogen. So we're looking at an AT pair. Based on the position of the methyl group, which side, left or right, is the T? Left. It's got the methyl group. That means this side has to be A. Okay? So I want you to be able to look at a space-filling model of double-stranded DNA and pick out and read the sequence. And it's just a matter of knowing what the major and major groove looks like and what functional group should be displayed there. You can do this with the minor groove too. The minor groove's more difficult to see though. It's narrower and deeper. Organic chemists have designed molecules that bind specific functional groups in the minor groove, but that's a story for another time and probably an entirely different class. Okay, let's do the one right above it. Take a minute, go look at your notes and see if you can figure out what the base pair, which I will now highlight here, is. And figure out which one's on the left and which one's on the right. Okay, got it yet? I see an oxygen. I see a ring nitrogen. And then I see an exocyclic amino group. So what does that look like? GC? Okay, it's a pretty good bet because it lacks a methyl group, right? If this is DNA, it doesn't have a methyl group, it's probably a GC pair. Oh, that's a secret, that's a trick. But yeah. And then how do you know which side is C and which side is G? The oxygen side is the G side, right? Yeah, because guanine, if we scroll up to the guanine GC base pair, C only has, the major groove side of C only has the exocyclic amine. The major groove side of G has both the ring nitrogen and the oxygen. Okay. So I can pretty well guarantee you a test question on that. And it requires you to know the structures of the bases, but also be familiar with Watson-Crick base pairing, but also to know major minor groove, and also to think about functional groups. So from an OChem perspective, I feel like it's a really nice question. Go ahead.
How do you know if it's a ring nitrogen or exocyclic?
Good. How do you know whether it's a ring nitrogen or an exocyclic nitrogen? Anybody want to answer that? Yeah.
One has hydrogen, so the other one looks attached to just carbon.
That's right. That's it. It's as simple as that. The exocyclic nitrogen will have NH bonds. So you'll see white hydrogens on the blue nitrogens. The ring nitrogen will just be a nitrogen attached to carbons. Good. Okay. All right, good. So you've seen the sugar phosphate backbone of DNA. I want to show you just a little bit about nucleotide versus nucleoside nomenclature. And then I want to show you how just an example of a reaction in oligonucleotide synthesis. We're just scratching the surface of DNA, RNA. We're leaving a lot to your biochemistry classes and MM Bio. There's more we could do, but I think this will be enough. So if you have the base plus the sugar, that's what we call a nucleoside. And there's really no favored pronunciation. There's a couple of different options. You will hear people say nucleoside versus nucleoside, doesn't really matter. But nobody ever really says nucleoside, so you got to pick one or the other anyway. I'll probably say it wrong now. Now I'm going to give myself a complex. So the base and the sugar is just the nucleoside. So the sugar is D-ribose, and the atoms in the ribose ring are numbered 1' at the anomeric carbon. There we go. Nope. So the anomeric carbon is 1', then 2', 3', 4', and then there's a hydroxymethyl group on the 4' carbon. That's the CH2 of that is the 5' carbon. 1' is the anomeric carbon. And this is the carbon that's attached to the nitrogen of the base. The linkage between the nitrogen of the base and the anomeric carbon, that's sometimes called an N-glycoside, meaning it's an acetal with an N instead of an NR instead of an OR attached to the anomeric carbon. All right, so we'll just do A, which has just the exocyclic amino group. So if you have the D-ribose attached to adenine, together the nucleoside name is adenosine. And as we did before with carbohydrates, this linkage between the anomeric carbon and the base, you can call that a glycosidic linkage. So naming nucleosides is a little bit different for the purine bases versus the pyrimidine bases. If it's A, as I said before, it's adenosine. If it's G, it's guanosine. And the idea is that the ose in there tells you that there's a sugar. So far, so good, makes sense. For the pyrimidines, we dropped the ose part and we named them a little bit different. So for U, it's uridine. For T, it's thymidine. And for C, it's cytidine. And that ending sort of matches the idine ending of the pyrimidine. So for the names for the nucleoside, pyrimidine nucleosides, you don't have really much clue that a sugar is there other than the name has changed from thymine to thymidine, from cytosine to cytidine, and from uracil to uridine. And this is why I said the naming of cytosine versus cytidine is confusing. You hear cytosine and you think, oh, there's a sugar there. For cytosine, it's just the name cytosine refers just to the base. The name cytidine refers to base plus sugar. All right. Now, there are deoxynucleosides that are precursors to DNA. For the deoxynucleosides, we're simply going to remove the 2' hydroxyl group. And there are enzymes that remove that 2' hydroxyl group. And we just add the word deoxy in front of the adenosine name. Deoxynucleosides will be a thing for both A and G, and they'll also be a thing for T and C, but not for U. You see what I'm trying to do here? It's not working, so let's just try this. We'll erase the uridine because uracil's found in RNA but not DNA. To abbreviate the deoxynucleosides, you just put a D in front of the letter, and then to write out the name, of course, you put the word deoxy right in front of the name of the nucleoside. Sorry about those double arrows. My copy and pasting is not going very well today. We'll just erase them. Okay. So you can recognize a deoxynucleoside from a nucleoside just by the presence of the 2' OH. Probably already knew that, but there it is. We won't talk about the enzyme that removes the OH group to convert a nucleoside into a nucleotide, or a nucleoside into a deoxynucleoside. All right, and then finally on the nomenclature list of things to cover, nucleotides are going to have the base, T stands for 3, the base, the sugar, and then it's going to have 1, 2, or 3 phosphates between 1 and 3. And when the phosphate is there, that's when it becomes a nucleotide. So ribonucleotides are going to be based on ribose. They're going to be exactly the same as you've seen here. I'm going to copy that and paste it here. The difference will be that the 5', that one of the OH groups is going to have a phosphate on it. And we'll name the nucleotide based on where the phosphate is. I'm drawing for you now the structure of ATP, which its systematic name would be adenosine 5' triphosphate. The 5' tells you that the 3 phosphates are attached at the 5' hydroxyl group. We specify that because there are some other nucleotides that have phosphate groups in other places. And we won't really worry about those or encounter them in this class, but you hear about them in biochemistry and some other areas, and so we'll put that there. This is a ribonucleotide because it's based on ribose. We would also abbreviate this as ATP. For the nucleotide triphosphates, we sort of count the phosphates away from the 5' hydroxyl with the Greek letters alpha, beta, and gamma. And you'll notice that the bonds between the alpha and beta phosphates and between the gamma and beta phosphates are a particular kind of functional group which are called phosphoanhydrides. Pretty reactive, as they're up at the top of our list of reactive carbonyl-like molecules. They're as reactive as acid chlorides or anhydrides, or as reactive as thioesters. They're just about as reactive as ketones and aldehydes. And so that reactivity plays a role in their chemistry, which we'll see a lot in metabolism. If we take the nucleotide triphosphate and we erase the 2' hydroxyl group, that becomes deoxyadenosine 5' triphosphate, or DATP. And when nature goes to make DNA and it needs to incorporate an adenine base at a specific spot, it uses DATP as the electrophile. All right, questions about nucleosides versus nucleotides? All right, in the last little bit, I just want to show you the chemistry for biological oligonucleotide synthesis. And we'll imagine that this is RNA. No, I take it back. Let's do DNA. So I'm going to copy my structure of DATP. This is going to be my electrophile. And I'm going to draw as my nucleophile the first nucleotide in this sequence. What we're doing is just an example. This would take place in the context of an enzyme. There would be a coding strand. There would be an already existing strand. In fact, we're going to put an OR on this 5' phosphate group to indicate that this is probably a much larger strand. Here's the 5' phosphate group. Here's the 3' hydroxyl group. That's going to be our nucleophile. And let's suppose that the previous base was a guanine, okay? And we want to make a new bond. Suppose the coding strand tells us we need to incorporate an A after this G. Okay, the alcohol is going to be our nucleophile, and one of the phosphoanhydride bonds of this DATP, this deoxynucleoside triphosphate, is going to be the electrophile. Now, I'll highlight the portion that's going to be the leaving group. It turns out that these triphosphates can be attacked at generally either the gamma phosphate or the alpha phosphate. And we'll see examples in metabolism of both of those. In DNA synthesis, we want to make a new bond between this oxygen and the alpha phosphate. So we're going to want to attack the alpha phosphate, and what's in green is going to be a leaving group. And if you look at that, that leaving group actually looks familiar. It's the pyrophosphate group that you saw before in terpene chemistry. And normally there's going to be a magnesium present to help neutralize some of the charge. What happens is an enzyme base will deprotonate the alcohol oxygen, and then the alcohol oxygen will attack the phosphorus, the alpha phosphorus. Move that alpha here. There are a couple of ways you can show this mechanism, and the way to show it is going to depend on whether the person showing it to you is in a hurry. It'll depend on the textbook and the author. It's tempting to just show this mechanism as SN2, attack the phosphorus, pyrophosphate leaving group leaves. And that's what a lot of texts will show you. There's at least some evidence that phosphate groups behave kind of like carbonyls. And we've used that analogy. We've talked about phosphoesters versus phosphoanhydrides. Well, with carbonyls, the chemistry isn't SN2, it's addition then elimination. You get an intermediate. And there's some evidence that that's going on here. So we attack the phosphorus, then we make a new oxygen phosphorus bond, and then the electrons kick up to form an intermediate, which I'll draw here. That intermediate is not a tetrahedral intermediate because it's got 5 things attached to it, not 3. By the way, I'm abbreviating the green stuff as the pyrophosphate group as we've done previously. Here is the oxygen that just came in from our ribose sugar, deoxyribose sugar. Here is the G base. Here is the 5' phosphate from that previous sugar. I needed a little bit more space, didn't I? And then this is the second ribose ring with the adenine base on it. Here is the alpha phosphorus. Do you see how what we've drawn here is kind of like the tetrahedral intermediate from carbonyl chemistry, only it's got 5 things attached to it. Then it's thought that the electrons on the oxygen kick down, leaving group leaves, and then what should be left after that is the new phosphodiester linkage between the G and the A. Oh, brother.
Isn't there supposed to be oxygen between the 5' and the phosphorus?
Yep, there is. Thank you. Sorry for that. That's better. Yuck. That's what happens when you're in a hurry. Okay, so that's how a single reaction elongating an oligonucleotide goes. Notice that this functional group here is called a phosphodiester because there's 2 OR groups attached to the phosphorus. Notice that the phosphodiester backbone of this oligonucleotide is negatively charged. And so when you look at double helix DNA, you now have an explanation for why the sugar phosphate groups are on the outside, because you've got the negatively charged phosphate groups and the polar groups on the sugar. And you've got the neutral, more hydrophobic bases, still pretty polar, but more nonpolar than the sugar and the phosphate on the interior. Okay, that's just about going to do it on DNA structure for us. So we will see you next time on Wednesday, where we will jump into chapter 27 on metabolism.